this lecture we are going to discuss two techniques. One is infrared spectroscopy and next one is fluorescence spectroscopy. Now, infrared spectroscopy is an example of absorption spectroscopy, whereas fluorescence spectroscopy is an example of emission spectroscopy. Now, infrared spectroscopy which falls under absorption spectroscopy, here transitions which are taken into account are the ones which happens between the vibrational labels in the ground state. That is, when a particular molecule or atom absorbs energy equivalent to the differences in the two vibrational labels, then they will absorb that particular form of energy. So, infrared spectroscopy exploits the fact that molecules have specific frequencies at which they rotate or vibrate. Now, when we consider molecules and they are like bonds between these molecules, these are they act like a spring actually, which can stretch, bend or rotate a bit around. They can rotate and produce differences, or you can say there is a constant movement of these atoms around these bonds, and they are either vibrating due to certain absorptions, and these transitions are measured in IR spectroscopy. So, if we have to determine if some compound is IR active or not, then if you consider for example, carbon dioxide, then carbon dioxide has two oxygens which are double bonded to the central carbon. So, here dipole moment from the symmetric stretch of the left oxygen will be offset by the symmetric stretch of the right oxygen, and therefore, it is considered as IR inactive. So, if you can see here, there are like stretches which are in offset by each other, which like and there could be different kinds of stretches in here. Now, in very simple terms, infrared spectroscopy can be defined as absorption measurement of different IR frequencies by a sample which is positioned in the path of an IR beam. So, if a sample is put in IR beam, there will be absorptional, and as I said, there will be transition will be measured in vibrational levels of ground state. Now, the photon energy which is associated with this part of the infrared spectrum is around 1 to 15 kilocalorie per mole, and it is not it is like only you can say large enough to excite electrons and induce vibrational excitation of covalently bonded atoms and groups. Now, the covalent bonds in the molecules, these are not rigid sticks, these are not like static here, but rather they could be in molecular model terms they are mostly like springs actually, or you can say stiff springs, and these could be stretched, bent, and other motions could happen in there. The main goal of IR spectroscopic analysis is to determine the chemical functional groups in the sample. So, different chemical groups like when IR spectra is taken, and in the sample if there are different types of chemical groups, each group depending on its chemical environment, conformation and chemical structure, and chemical environment means environment around it like solvents and other parameters that will affect its motions like stretching or bending, and that particular characteristic feature of these every type of bonds will give a characteristic spectra which is very helpful in determining these chemical functional groups in a particular sample or particular molecule. Now, using various sampling accessories, IR spectrometers can accept a wide range of sample types, which could be range from gases, liquids and solids, and there are certain limitations also for these. So, IR spectroscopy is a very important and very popular tool for structural elucidation and compound identification. Now, here when we say structural elucidation, it does not mean 3-dimensional structure, but it will tell about the functional groups, it will tell about also IR spectroscopy like FTIR spectroscopy can tell about secondary structural information and also changes in structure like say in different environmental conditions. If we consider IR frequency range or the spectrum here, it lies between the microwave and visible parts of the electromagnetic spectrum. This we have discussed in the last chapter actually. Now, far IR has low energy and may be used for rotational spectroscopy, and mid IR may be used to study the fundamental vibrations and associated rotational vibrational structure. Then there is near IR can excite overtone or harmonic vibrations. So, if you can see here, this is the spectrum in electromagnetic spectrum. Infrared lies between visible, that is near infrared, and then this is far infrared, and this is the between the microwave and visible regions actually. Also like here, if you can see, this lies between 10 raised to power minus 4 to around here 10 raised to power minus 2 centimeters, or you can measure it in different forms actually. Now, infrared radiations spans a section of the electromagnetic spectrum and have wave numbers from roughly 13,000 to 10 per centimeter. Now, wave number is inverse of wavelength actually, and that is the most commonly used in here, or frequency is utilized here to plot absorption spectrum. So, or we can say it in terms of wavelengths is around 0.78 to 1000 micrometer. Now, it is bound by the red end of the visible region at high frequencies and the microwave region at low frequencies. So, IR absorption positions are generally presented as either like I said, it is mostly presented as wave numbers rather than wavelengths. Now, wave number defines the number of waves per unit length actually. So, thus wave number are directly proportional to frequency as well as the energy of the IR absorption. The wave number unit, that is per centimeter or reciprocal centimeter, is more commonly used in modern IR instruments that are linear in the per centimeter scale. In contrast, wavelengths could be used which are inversely proportional to the frequency and their associated energies. Wave numbers and wavelengths can be interconverted, like wave number is in reciprocal centimeter and wavelength will be in centimeter actually. So, that way you can convert that. Now, IR absorption information is generally presented in the form of a spectrum with wave number as on the x axis and absorption intensity or transmittance as the y axis, like in UV visible also we have seen its absorption is on the y axis as I have shown you, and the wavelength or here wave number is on x axis. And also transmittance could be taken, which is the ratio of the radiant power transmitted by the sample to the radiant power incident on the sample, as we have seen in the Beer's law in UV visible spectroscopy. So, here also same principles applies. Now, the transmittance spectra provide better contrast between intensities of strong and weak bands, because transmittance ranges from 0 to 100%, whereas absorbance ranges from infinity to 0. So, one should be aware about, or the one who is doing analysis should be aware that the same sample will give quite different profiles for the IR spectrum, which might be linear in wave number, and IR plot which is linear in wavelength. So, that has to be taken into account. So, it will appear as if some IR bands have been contracted or expanded if you compare the wave number spectrum to wavelength spectrum, and the frequency used in mid IR region is around between like 4000 to 400 per centimeter or 2.5 to 25 micrometer. The far IR requires the use of specialized optical material and sources. It is used for analysis of say organic, inorganic and organometallic compounds involving heavy atoms. It provides useful information to structural studies such as the conformation and lattice dynamics of samples. Near IR spectroscopy needs minimal or no sample preparation. It is very typical. It offers high speed quantitative analysis without consumption or destruction of the sample, and the instrument can often be combined with UV vis and coupled with fiber optics device for remote analysis. Near IR spectroscopy has gained quite increased interest as particularly in process control applications. Now, if we consider the theory of infrared absorption here, I think all of you are aware that at very low temperatures, that is temperatures above absolute 0, all the atoms in the molecules will be like at any temperature for that matter. If you are above a particular like lowest temperature, that is absolute 0, then atoms or the molecules they are not static, they are in continuous vibration with respect to each other, and their vibration can be in different directions actually and in different ways. So, when the frequency of a particular specific vibration is equal to the frequency of the IR radiation directed on the molecule absorbs the radiation, and each atom has here 3 degrees of freedom actually corresponding to motions along any of the 3 Cartesian coordinates, that is x, y and z. Now, major types of molecular vibrations that will be there are stretching and bending, and there could be others also, more complex one, and infrared radiation is absorbed and the associated energy is converted into these types of motions. So, that is a continuous process which keeps on happening. Absorption will involve discrete quantized energy levels like as we have seen in vibrational levels within the electronic level. However, the individual vibrational motion is usually accompanied by other rotational motions, and these combinations will lead to the absorption bands, not the discrete lines commonly observed in mid IR region. Like we have discussed earlier also that there are a lot of discrete lines, but these are closely spaced lines which appears as bands. In simple terms, if we say how it works actually, the IR spectra are obtained by detecting changes in transmittance or absorption intensity as a function of frequency, and most commercial instruments separate and measure IR radiation using either dispersive spectrometers or Fourier transform spectrometers. Now, dispersive spectrometers they were introduced in mid 40s and they are widely used spectrometers, and they provide the robust like these are instrumentation required for the extensive application of this technique. Spectrometer components, if we take the IR spectrometer, consists mainly of 3 basic components as in other spectrophotometer as we have discussed earlier. One should be like in any other spectrometer, one is radiation source, monochromator and detector, and then a place for sample to be put in. Now, the common radiation source for the IR spectrometer is an inert solid heated material that electrically to say from 1000 to 1800 degree Celsius. Now, 3 popular types of sources are these are Nernst Gloer constructed of rare earth oxides, then Glober constructed of silicon carbide, and there is nichrome coil actually. So, these are different radiation sources, and these all produce continuous radiations, but different radiation energy profiles. Then monochromator is a device used to disperse a broad spectrum of radiation and provide a continuous calibrated series of electromagnetic energy bands of determinable wavelengths or frequency range. Then prisms or gratings are the dispersive components used in conjunction with the variable slit mechanisms, mirrors and filters. For example, a grating rotates to focus a narrow band of frequencies in a mechanical slit. Narrower slits enable the instrument to better distinguish more closely spaced frequencies of radiations and will result in better resolution, whereas wider slits allows more light to reach the detector and provide better system sensitivity. So, there has to be a compromise in the settings for desired slit width and to get good results. Most detectors used in dispersive IR spectrometers can be categorized into two classes. One is thermal detectors and photon detectors. Now, thermal detectors will include thermocouples, thermistors or pneumatic devices, and they measure heating effects produced by infrared radiation. Other and photon detectors will measure photons actually. Now, basic IR spectroscopic setup here in this particular one, that IR source emits an IR beam, and IR beam here in very basic way, IR beam here is split into two identical beams. Now, one beam of the split beams goes through the sample and other will go through the reference cell. So, that is how it will occur. Now, reference cell typically consists of the solvent that sample is dissolved in, and IR used to measure the amount of energy absorbed when the frequency of the infrared light is varied. In pulsed Fourier transform IR, a single pulse is sent through the sample, and this will contain many frequencies which will allow for a much faster test. In a typical dispersive IR spectrometer, radiation from a broadband source passes through the sample and is dispersed by monochromator into component frequencies. Then the beams fall on the detector, which generates an electric signal and results in a recorder response. Most dispersive spectrometers have a double beam design, and two equivalent beams from the same source pass through the sample and reference chambers respectively. So, using an optical chopper such as sector mirror or other, the reference and sample beams are alternatively focused on the detector, and commonly the change of IR radiation intensity due to absorption by sample is detected as an off null signal that is translated into the recorder response through the actions of synchronous motors. So, this is a very simple schematic here, that you have an infrared source. These beams are then divided into two. Here one goes to the sample, another to reference, then finally it goes through the prism and to detector and recorder. As we have there are a lot of like filters and like prism and other monochromators which are put in here, as we have discussed just now. So, this is a very simple schematic of how the transmission occurs. As far as Fourier transform spectrometers are concerned, Fourier transform spectrometers have replaced not completely, dispersive instruments for many applications due to the superior speed and sensitivity, and they have greatly extended the capabilities of infrared spectroscopy and have been applied to many areas that are very difficult or near impossible to analyze by dispersive instruments. So, instead of viewing each component frequency sequentially, as in a dispersive IR spectrometer, all frequencies are examined simultaneously in FTIR spectroscopy. Two main forms of preparation for here sample preparation for solids begin with grinding the material to a fine powder and then dispersing it in a matrix. Potassium, mostly potassium bromide is the widely used matrix. The ground material can be dispersed in a liquid, usually like mineral oil or nuzol, to form a mull. Typically, no more than 20 milligram of solid is grounded, and then 1 or 2 drops of nuzol are used to create a paste, which is then spread between the two IR transparent windows. Then the mixture will be like transferred to a die that will have a barrel of a particular diameter, and they will be pressed, and finally at a particular pressure, and then material which is recrystallization of the KBr results in a clear glassy disc, which can be analyzed by transmission. Now, liquid preparation is easier than solids and is traditionally prepared as thin film cells, where the cell consists of IR transparent windows, and it is important to have constant path length for the laser to travel through and that the sample is homogeneous here. Now, it is possible to obtain an IR spectrum from samples in many different forms like liquids, solids and gases, but there are problems. Many materials like are opaque to IR radiation and must be dissolved or diluted in a transparent matrix to obtain spectra. Alternatively, it is possible to obtain reflectance or emission spectra from opaque samples. Then typically solutions in particular concentrations like 0.05 to 10% concentrations are handled in IR cells of 0.1 to 1 millimeter in thickness. Concentration of 10% and cell path length of 0.1 millimeter represents one practical combination. In a double beam spectrometer, compensating cell is filled with pure solvent and placed in the reference beam, as we have discussed. In the single beam instruments, the solvent bands are mostly removed by obtaining the difference spectra through subtraction of the solvent, that is zeroing of that particular spectra. Both fixed thickness and variable thickness liquid cells are available commercially and normally consist of metal frame plates. IR transmitting windows and gaskets that determine the path length of the cells, these are all available. Now, salt plates or IR transmitting materials can be used for semi volatile and non volatile liquid samples, like sodium chloride discs are the most popular and economical choice for this purpose. Silver chloride or barium fluoride plates may be also used for substances that will dissolve or react with NaCl plates. The sample preparation here is also in a similar way. A drop of the sample is squeezed between the two salt plates to form a film of very fine thickness like 0.01 millimeter or so. Then plates can be held together in a capillary attraction or may be clamped in a screw tightened holder, and then it is possible to place the film of samples on salt plates by melting a relatively low melting solid and squeezing it between two plates. The solid sodium chloride crystal salt plates can usually be cleaned with dry methylene chloride or acetone, and this smear technique is one of the simplest ways to obtain IR spectra. So, there are various methods, and like I said, like we discussed, like there are two important instruments here, dispersive and Fourier transform IR spectroscopy. There are a lot of applications of IR spectroscopy. It mostly gives much of analytical information which could be qualitative, that is the combination of the fundamental vibrations or the rotations of various functional groups and subtle interactions of these functional groups with other atoms of the molecule will result in the unique generally complex IR spectrum for each individual compound. So, these are characteristic spectra are obtained, and it could be utilized for structural elucidation and compound identification. In structural elucidation, because of the complex interactions of the atoms within the molecule, IR absorption of the functional groups may vary over a wide range. However, it has been found that many functional groups give characteristic IR absorption at specific narrow frequency ranges, regardless of their relationship with the rest of the molecules. So, this both ways it could happen, that spectra could be affected by the chemical environment, but in certain cases it might be as it is not affected so much. There are generalized tables of the positions and relative intensities of the absorption bands and have been established by certain groups. So, these these strong absorption bands or the characteristic spectra in a particular region of the, like say, wave numbers usually comes, like say, from 4000 to 2500 per centimeter, usually comes from stretching vibration between hydrogen and some other atoms with a mass with a particular mass which is less. The OH and NH stretching frequencies fall in 3700 to 2500 per centimeter. Then various intensities with various intensities. The hydrogen bonding has a significant influence on the peak shape and intensity. So, that is also very important. Then the CH stretching bands occur in region of 3300 to 2800 per centimeter, and there are various, like we can go on where different kinds of like aliphatic, saturated or double bond compounds, they have a characteristic spectra. Different regions could be taken. There are certain regions called fingerprint region from 1300 to 910 per centimeter, and absorption in this region includes the contribution from complex interacting vibrations, giving rise to generally unique fingerprint for each compound. A good match between the IR spectra of two compounds in all frequency ranges, particularly in the fingerprint region, will strongly indicate that they have the same molecular structure. So, that way the two molecules could be compared if they have same structure, or an unknown molecule if IR spectra matches to certain as per wave numbers, they could be also identified. So, there are a whole lot of tables or standard values to which the IR absorption in different regions can be correlated, and it could be found out. So, structure elucidation in, like I said, structures of say proteins, secondary structures could be determined. It could be complementary to circular dichroism technique, and this is because of differential hydrogen bonding patterns and their effect on IR spectrum which could be observed. The compound identification is another important application, and here since IR spectrum of every molecule is unique, and so identification methods often say organic compound or other compounds or say amide bond or particular functional groups can be performed through comparing it with large number of reference spectra in various conditions actually, and that way one could this database could be utilized for different areas in biotechnology, like forensics, biochemicals, biochemical analysis or polymer analysis, or even forensic science it could be utilized. There are computer search programs which can facilitate the matching process. In many cases where exact match to the spectrum of an unknown material cannot be found, they will list the reference compounds that match the unknown most closely. So, this kind of information is very, very important. Certainly, this is very useful information, and when it is combined with say NMR or mass spectrometry, positive identification with high confidence could be achieved. There could be quantitative information also, where IR spectroscopy mostly was considered qualitative, but quantitative and semi quantitative analysis it is considered as qualitative and semi quantitative only. but like this could also be like with FTIR instrumentation and strong computerized data processing capabilities have greatly improved the performance of this quantitative IR work, and this modern infrared spectroscopy has gained acceptance as a reliable tool for quantitative analysis also. The basis for quantitative analysis of absorption spectrometry is again the Beer's law, which is Beer-Lambert law, and for a single compound or homogeneous medium, it will be expressed as we have done, where there will be a molecular absorptivity, path length, and the concentration multiplied to each other gives the absorbance. So, law basically states that the intensities of absorption bands are linearly proportional to the concentration of each component in a homogeneous mixture or solution, and deviations of Beer's law occur more often in infrared spectroscopy than UV vis, but still it is a way to measure or to quantitate particular material. So, like we have discussed, these applications of IR spectroscopy can be in various areas of biology and other areas, where like we have seen that different kinds of analysis, right from identification of functional groups or structural information about molecules in different forms could be derived from IR spectroscopy. Even titrations like pH titrations could be performed. Hydrogen bonding in different environments could affect IR spectroscopy, and that could give structural information. Tautomeric groups could be identified. Then there is lot of applications in like other industries like petroleum, oil and or grease, like to analyze these contents and other applications. It could be also applied, like I said, in forensics or biochemistry or other areas. It could be also have lot of applications. So, IR spectroscopy is one very important analytical tool which could be applied, which have applications in the field of in biotechnology for various kinds of to gain information for various kinds of problems actually, and it could be combined with other techniques like NMR or mass spectrometry. So, this completes the section on IR spectroscopy. Let us move on to the next one, that is fluorescence spectroscopy. Now, as we have discussed about earlier for UV vis, that is a absorption spectroscopy, but fluorescence spectroscopy is a emission spectroscopy. It is complementary to the absorption spectroscopy. Now, here molecules have like as we say various states which are energy labels actually. In fluorescence spectroscopy, what is done is like there is like excitation. It is concerned primarily with electronic and vibrational states, as we have shown in the energy diagram. Let me show you here a very simple way the principle of fluorescence spectroscopy. So, if you could recall the energy diagrams which is which we have made in these forms. Now, if you could recall, there were vibrational labels in here, different vibrational labels, and this is the ground state and this is the first excited state. Now, these are the electronic state and these ones which are thin lines are vibrational states. So, what happens is there is an excitation which occurs. Now, in absorbance this will be like a different thing, but in fluorescence what happens is the excited atom or electron will first fall through these vibrational labels here, and then it will come down to a particular vibrational label in a ground state, but it comes to higher to a different label where there is certain emission of certain photons, which the phenomenon is known as fluorescence. All right. So, in very simple terms we have tried to understand the phenomenon of fluorescence. Uh, so, here what is happened, that species being examined has a ground electronic state, that is a low energy state of interest, and an excited electronic state of higher energy. In fluorescence spectroscopy, the species is first excited, as I have shown you, by absorbing a photon from its ground electronic state to one of the various vibrational states in the excited electronic state, and then with it comes back like through different interactions, maybe uh, it will reach the lowest vibrational state of the excited electronic state, and then molecules may drop down into any of the several vibrational labels in the ground state. Now, the emitted photons will have different energy and thus frequencies, and therefore, by analyzing the different frequencies of light emitted in fluorescent spectroscopy along with their relative intensities, the structure of different vibrational labels can be determined. Now, fluorescence is an emission phenomenon rather than absorption, where an energy transition from a higher to lower state is accompanied by radiation. Only molecules in their excited forms are able to emit fluorescence, and thus they have to be brought into a state of higher energy prior to the emission from phenomenon. So, if there is a chromophore or there is a fluorescing material, first radiation for absorption has to be provided or it should be excited. So, here if we can see here the fluorescence in this case, if these are going from ground state to one of the vibrational state, and when they come back the one which is not like when they come back they will emit certain photons like in here, and they will show the fluorescence. So, only those which have a difference in energy will show the fluorescence, not all of them when they are excited and when they come to ground state will show fluorescence, but only certain compounds will show fluorescence. The fluorescence properties of a molecule are determined by properties of the molecule of itself, that is internal factors, as well as the environment of the particular, say if we consider protein, then external factors. The fluorescence intensities emitted by a molecule is dependent on the lifetime of the excited state. So, if we consider protein, the fluorescence properties will be determined by the properties of the molecule as well as the environment of the protein as well. So, the transition from the excited to the ground state can be treated like a decay process of first order, that is number of molecules in the excited state decrease exponentially with time, and it could be like it like analogy to kinetics, where exponential coefficient which is called rate constant is the reciprocal of the lifetime. The lifetime is the time it takes to reduce the number of fluorescence emitting molecules to a particular label, and they are proportional to lambda cube. Now, the ratio of photons emitted and photons absorbed by a fluorophore is called quantum yield actually, and the quantum yield is a dimensionless quantity, and most importantly the only absolute measure of fluorescence of a molecule. Measuring the quantum yield is a difficult process and requires comparison with the fluorophore of known quantum yield. In biochemical applications, the measurements is rarely done, and most commonly the fluorescence emissions of two or more related samples are compared and their relative differences are analyzed. So, this was a little bit about the principle, or you can say theory of fluorescence. Instrumentation, if we consider, there are in general two types of instruments exist, which are filter fluorometers, which use filters to isolate the incident light and fluorescent light, and there are spectrofluorometers which use diffraction gratings monochromators to isolate the incident light and the fluorescence light. Now, both types use the following the scheme where light from an excitation source passes through a filter or monochromator and strikes the sample. Now, a proportion of the incident light is absorbed by the sample and some of the molecules in the sample will fluoresce. So, remember you have to have a fluorescing compound in the sample, which could be intrinsic or extrinsic. Intrinsic means inherent property of the molecule to fluoresce, and extrinsic means you have put in a dye or other fluorescing material attached to the sample. Now, the fluorescent light is emitted in all directions, and light also passes through a second filter or a monochromator and reaches detector, and this is usually placed at 90 degrees to the incident light to minimize the risk of transmitted or reflected incident light reaching the detector. Now, various kinds of light sources may be used, excitation source including lasers, photodiodes and lamps like xenon arcs and mercury vapor lamps are in particularly used. Laser only emits light of high irradiance at a very narrow wavelength interval, typically under 0.01 nanometer, which makes an excitation monochromator or filter unnecessary here. Disadvantage of this method is that the wavelength of a laser cannot be changed much. A mercury vapor lamp is a line lamp, meaning it emits light near peak wavelengths. By contrast, a xenon arc has a continuous emission spectrum with nearly constant intensities in the range say from 300 to 800 nanometer and sufficient irradiance for measurements down to just above 200 nanometer. So, different kinds of sources could be utilized in here, and which we also discussed in fluorescence microscopy some of these things. Filters or monochromators may be used in fluorimeters. Like I said, a monochromator transmits light of an adjustable wavelength with an adjustable tolerance, and the most common type of monochromator utilized is a diffraction grating, that is collimated light illuminates a grating and exits with a different angle depending on the wavelength. The monochromator can then be adjusted to select which wavelength to transmit, and for allowing anisotropy measurements there could be also addition of polarizers or polarization filters. One alters the excitation monochromator and one before the emission monochromator or filter. So, that is fluorescence polarization could be achieved in there. The fluorescence is most often measured at 90 degree in spectrophotometers. If you could recall, in microscopy it was not so relative. So, here it is at 90 degree to relative to the excitation light, and the geometry is like here this geometry is used instead of placing the sensor at the line of excitation at 180 degree angle. So, there could be different kinds of arrangements in here. Now, no monochromator is perfect and will transmit some stray light, that is light with other wavelengths than the targeted. Further, the fluorescence can also be measured from the front, which is often done for say opaque or turbid samples. Then the detector can either be here single channeled or multi channeled. The single channel detector can only detect the intensity of one wavelength at a time, while the multi channel detectors can detect the intensity at all wavelengths simultaneously, making the emission monochromator or filter unnecessary. So, the different types of detectors both have advantages and disadvantages, and also in terms of cost factors. So, that could be chosen as per requirement here. The most versatile fluorimeters with dual monochromators and a continuous excitation light source can record both an excitation spectrum and a fluorescence spectrum. So, when measuring fluorescence spectra, the wavelength of the excitation light is kept constant, preferably at a wavelength of high absorption, and the emission monochromator scans the spectrum. For measuring excitation spectra, the wavelength passing through the emission filter or monochromator is kept constant, and the excitation monochromator is scanning. So, the excitation spectrum generally is identical to the absorption spectrum, as the fluorescence intensity is proportional to the absorption. So, there could be different arrangements in here. This is a very simple schematic of spectrophotometer. Uh, there is a lamp here and there is a grating slit, there is a mirror which reflects light to reference detector, and there is a sample cuvette also. So, both will like through again grating slit, and there is a excitation filter and emission filters here, and finally, data is collected on a sample detector and then sent to the computer. So, this is like as spectrophotometers are. This is a here detection is at 90 degree as opposed to in a straight line. So, fluorescence spectroscopy uh, is a very useful technique and works most accurately at a very low concentration of emitting fluorophores. The better fluorescence spectrometers in laboratories will have a photo counting detector yielding a very high sensitivity. Fluorescence is a very sensitive technique, and there should be temperature control is required for accurate work, as the emission intensity of fluorophore is dependent on the temperature. So, that is also very important for spectrophlorimeter as well as for other works also. The temperature control is a very important. This is a very there is a schematic of fluorescence spectrophotometer as we have shown earlier. So, you can see like it starts from here, there is a light source, there is a dual grating excitation monochromator, the shutters, filter holders, beam splitters, and then there is a sample chamber here, then there is a emission monochromator, and finally, photomultiplier for detection. So, it contains in essence it contains both excitation as well as emission monochromators, sample holder. It for say polarization effects, it would have two polarizers as well attached to it. So, there could be fluorescence polarization could be observed. Now, there are a lot of applications of fluorescence spectroscopy in biotechnology and otherwise. Fluorescence spectroscopy is used for biochemical, medical, chemical research, and various other fields for analyzing say organic compounds and a lot of other compounds. Then there is like also has been there are reports which is used in differentiating malignant skin tumors from benign. Atomic fluorescence spectroscopy techniques are useful in other kinds of analysis measurements of compounds present in air, water or other media. Like heavy metals detection could be performed like mercury. Fluorescence can be used for to redirect photons. There are many and highly varied applications for fluorescence. Despite there is a problem here that in biology few compounds exhibit the phenomenon of fluorescence, and so if there is a problem like say in proteins tryptophan exhibits that, so that could be used like riboflavin and others could exhibit, but again problem is there are not many of these. In that case, in extrinsic fluorescence or certain fluorescing compounds can be attached to these molecules and then they could be monitored. Effects of pH, solvent composition and the polarization of fluorescence may all contribute to structural elucidation. Non-fluorescent compounds like I said can be often labeled with fluorescent probes to enable monitoring of molecular events like following it in a cell, or for say structural information or certain chemical reactions. All these things could be utilized in here like exchanging of groups and others. So, extrinsic fluorescence is as distinct from intrinsic fluorescence, where you have a native compound exhibiting that property. Fluorescing dyes are sensitive to the presence of metal ions and these can be used to track changes of these ions in vitro samples. And if you could recall, we have discussed about many applications in microscopy of fluorescing dyes, like say for calcium detection and other lot of different ions could be detected in here. Proteins possess three intrinsic fluorophores, mainly the tryptophan. Tyrosine and phenylalanine have a very low quantum yield, particularly phenylalanine has very low quantum yield and doesn't contribute much to that. But usually tryptophan fluorescence can be done in 295 to 305 nanometer range. The main application of intrinsic protein fluorescence aims at conformational monitoring, that is conformational changes in different conditions or in various environments where say tryptophan is exposed or it is present or it is not exposed in the structure. Frequently molecules of interest for biochemical studies are non-fluorescent, and many of these cases, like we said, external fluorophores could be attached here. So, that could be coupled in different forms, like for example, green fluorescent protein which could be cloned with the protein of interest, and it could be used for fluorescence experiments. There are a lot of different extrinsic chromophores or fluorescing compounds. One like green fluorescent protein, there are a lot of dyes. Another one is ANS or 1-anilino 8-naphthalene sulfonate, which emits a weak fluorescence in polar environment, but when it combines with proteins say in hydrophobic environment, it fluoresces. So, those properties could be differential properties could be exploited. There are a lot of other, like we have discussed earlier, for calcium there are Fura2 or Endo1 dyes. There are intrinsic fluorescence for nucleic acid. If we consider, they are very weak and required excitation wavelengths are too far in the UV region for practical to be used for practical applications. So, numerous extrinsic fluorescent probes could be combined with DNA for enhanced emissions. So, as we have seen and we have discussed earlier also, fluorescence spectroscopy could be utilized for a lot of applications, like we have discussed in the microscopy sections, where it could be applied for localization or for like in spectroscopy it could be utilized for structural information, for localization, for exchange of groups, for quantitative or qualitative terms, knowing metal ions and their exchanges. So, there are a whole lot of applications. It is a very sensitive technique, and phenomenon of fluorescence is very quick, very fast, where decay rates, as we have discussed earlier, is very fast. So, this technique is another very important technique which is used in biotechnology for different applications. So, this completes two techniques here in this lecture. One is IR spectroscopy, another is fluorescence spectroscopy. One can always refer to literature and other for more information. These lectures we have tried that you can understand basic concepts and what could be the applications and use in different areas of biotechnology. Thank you.